It's great to see you all, and really want to welcome you to the ISAM Congress in Asia. This is our first time in 29 years. It was in Sendai the last time in 1997. So we're really excited to have you all here, and really want to recognize the people who helped put this on. Hui Ling, especially, and Wei Ren have really done a tremendous job. We're really excited to see the program taking shape. So really want to recognize that this Congress is going to be a new opportunity to reconnect with Asia for the ISAM Congress. Hui Ling has done a tremendous job in raising funds to support this, and she's brought together all of you and the people outside. And so now it's up to all of us to make sure that we're really engaged and active in this to make sure it is a success. So thank you everyone.
Thank you, Dr. Chipola. Next, it's our great pleasure to welcome President Ming Tang of Chang'an University to deliver his opening remark.
Hello everyone, I'm the president of Chang'an University. Some of you may be wondering, what is Chang'an University? You may not know this university, but I believe that all of you must know there is a Chang'an Memorial Hospital, which is the largest hospital chain in Taiwan. We share the same name because we have the same founder, Wang Yongqing. He was the chairman of the Formosa Plastic Group. He founded one hospital chain and 3 universities. And we are honored to be the host by International Society for Aerosols in Medicine to have a conference here. In all the academic career life, we always go to conferences. I remember when I was young, when I was a assistant professor at the University of Illinois, Champaign-Urbana, I went to the conference. I listened to good ideas. I said, hey, how come I'm so down? How come I couldn't come up with this idea? So look at other scholars' presentation. I say, gee, I have to go back and work harder. But also in the conferences, you always receive stimulations from other participants. I think it was very horrible in the Taoyuan airport. So welcome to explore Taipei, and welcome to this conference hosted by Chang'an University. I wish this conference will be a great success. Thank you.
Nucleic acid or vaccines. As far as I know, that the recent progress in aerosol medicine that include the nanoparticle formulation for data absorption, and also include the room temperature.
This, as you can know, we are very focusing on the pulmonary and also some of the aerosol and also some of the respiratory system, disease, cooperation and relationship with all of you in the world. In this way, I hope we can do some more and more help to the patient and to the population. Welcome to Taipei again. I think Taipei is a very warm city, warm people here, and especially you can try the delicious food, just like my style. Okay, thank you again. Welcome. Thank you.
Between ERS and the people around the eastern part of the world. We very much would love to it. And I would like to invite you to ERS congresses this year in Barcelona and next year in Milan, and hope to see you here. Thank you. Thank you very much.
It's my first time in Taiwan, and I'm really happy to be here. I had a couple of days already to enjoy the food and to enjoy some of the sights, but I'm looking forward to the week more and see Taipei and talk to you and meet you all.
What can I say in addition to David and to Sabina? But honestly, ISAM is really unique globally. It's got a very unique USP of translational science and observations at the bench that cross-cut, and also aspects related actually get the science first for investors, for researchers, for academia, for clinicians.
And in the end, I would like to say thank you for the Taipei University, and thank you for the meeting, for supporting and to organize this conference. I welcome you. Thank you. Enjoy days in Taipei.
Good afternoon, distinguished guests and colleagues and friends. It's on behalf of the organizing committee, it's my great honor and pleasure to welcome you to the 26th ISAM Congress here in Taipei. Your presence today is truly appreciated, and we are especially grateful to those who travel around the world through the travels, and your determination to be here reflects strength and spirit of our global ISAM community. And at this point, we still have two guest speakers coming in on their way. And also, we have one speaker who cannot be here with us due to the medical illness, and she is determined to stay at 3 a.m. her time to present for us this later afternoon. So I'm the one really touched by all your determination and spirit and all your efforts, and I really appreciate. And it's really, it's especially meaningful that ISAM returned to Asia after nearly 3 decades. This year, we are delighted to welcome participants from 70 organizations from across 20 countries, bringing together researchers, clinicians, engineering, and industrial leaders, and with a shared goal to advance aerosol science and improve patient care. Over the next 4 days, we have prepared an exciting program and featuring 2 hands-on network workshop, featuring by representing the 6 our network groups, also 6 sections and reflect to our network groups, also and 3 flash talks and 2 oral presentations. So you will see another program and a busy schedule. And together, those sections focus the latest advances in inhaled drug delivery, respiratory therapeutics, and medical devices, and the translational research. And this program, as Omar was mentioned, the ISAM is really open and vibrant with all discussion and collaborations. So the science program is built through the collaborations across disciplines, across borders. I really encourage you to use these 4 days to actively participate, exchange ideas, and connect with colleagues throughout the Congress, especially for our students and early career investigators, which represent a future of our field. And finally, I would like to express my sincere appreciation to our invited speakers and chairs, sponsors, volunteers, and entire organizing committee for making the ISAM 2026 possible.
Care medicine at China Medical University Hospital. He also served as a professor in the School of Medicine at China Medical University. Dr. Han received his MD and PhD degrees from China Medical University, and Dr. Han served right now was the president of the Taiwanese Sleep Medicine Society, playing a key role in establishing a formal system for sleep medicine in Taiwan. And now currently he has various clinical trial on respiratory medicines and anti-inflammatory drugs. And today he is going to present the inhaled therapy in pulmonary fibrosis.
Okay, thanks the society inviting me to this conference. This is my first time to talk about aerosol therapy. I'm not an expert. As the moderator said, my special focus on
Kaohsiung Matistan Vincent Hospital, and he received his and PhD degree from the National Taiwan University. And today he's going to introduce the topic.
Thank you, Professor Lin and Professor Silva for your nice introduction, and thank you for the organizing committees to
where the unmet need and where the gap is, and the physician's perspective for how to solve the gap. So that's why we have two very unique physicians here to grab in some perspectives for their for the future possibility of future research. And now we open to the floor for one question. Have shared outstanding science, inspiring discussions, and innovation ideas. More importantly, we have strengthened cooperation. I hope we all do. Build new friendship and welcome the next generation of patient, science, and clinician into our community. And we hope you have enjoyed not only the science program, but also the hospitality and culture of Taiwan. And although the Congress is ended and the collaboration ideas that begin here will continue to grow to and advance aerosol science for the benefit of patients around the world.
The exhibitor, attendee, and speaker. And so we can't make it without anyone here. So hopefully enjoy the Taipei and the ISAM Congress this year, and hope we can, like Hui-Ling just mentioned, hope like everyone could like have different type of collaboration and have most importantly friendship. This is I think this is very special for me because I can gather invite my formal advisor and everyone to come to this room together. Yeah, so hopefully we will meet next year in Dublin. Yeah, definitely will go. So yeah, custom. See you next year. Yeah.
Next, I really sincerely would like another applause for our event company, for all the staff from the event company. You've seen me running around throughout the whole floor, and without them, we could not be well and be happy throughout the event. And finally, on behalf of the organizing committee, I thank you again for being here, and we wish you a safe journey home, and look forward to seeing you at 2027 ISAM Congress in Dublin.
Then use it in animals to see where your drug went? You don't know. So this was the first step to establish that when we had a particle size of 2 microns that was not with tracer, it stayed the same with the tracer. Yeah? And so this is had been lavaged to get their surfactant out to simulate respiratory disease. Rob's actually figured out a chimera model where he has a 28-week. You can see this is actually his nasal upper airway for the 28-week-old, and he attaches it to the trachea of the animal, and the animal can actually normally ventilate through it. Because rabbits' upper airways are very different from babies. So this allowed us to then do some scintigraphy and figure out how much got into the buddy. Yeah? And what we found was of the total inhaled dose, about 60% was inhaled, with about 18% in the upper model, with about 42% in the lung. Well, that's great, because that's exactly what we got with our in vitro model. So now we have in vivo correlation, right? The next thing that we had to do was one question: did you dilute the surfactant for aerosol?
No, I didn't dilute it.
Because one of the things with the mesh, people reported that if they dilute the saline, they more than double the rate of nebulization because it's so viscous.
Yes, but we can see the video I showed before. The mist can generate smoothly, so I didn't
very much. So this is our time for our partner showcase. And while we enjoy all of our sponsors and appreciate them for the support that they give our conference and the information they share with their presentations at the booths, we really like people who come with data and are willing to share it with them. So we have 3 presentations. They're going to run, we've got 10-15 minutes because we have a little bit extra time to play with questions and answers. So first we're going to ask Anna Costa to come up. She's with Fluida and the group in Conchette, Belgium. Yeah? He.
Everyone come. I hear Omar yelling in the lobby. It's good. Anyway, we hope that you'll pay attention because your vote will count for the best presentation. We will show a QR code at the end so that you can vote for the best presentation. So please pay attention for our speakers. First up, our speaker is, let's see.
But, okay, we have the targets. We know the type of molecules, so therapies and the molecules. Now, what else are they? We should be looking at mechanisms of action. Essentially, there are 2 categories. You're either an agonist or you're going to open channels. And this is the major path for targets. So essentially, if you only need to remember 2 things here, you're either going to be triggering receptors or you're going to open ion channels. These are the 2 main categories for what's in development in phase 2 and 3.
The private healthcare insurance, where you are directly spending your money, you see what you get, right? And what you spend for, and then you can give that to your healthcare insurance, the invoice and everything. If you have the standard healthcare insurance by the state, you don't see at all what it costs and what the physician has actually done with you or what they're putting on the bill. So, but if you come from the private insurance side, you know that it costs money and that you are spending this, and maybe the access is easier then because generally there's a different access to it.
Maybe you're right. Maybe we need to go to the side.
If there are no further questions, I would say thank you, Sabina.
Thank you.
Thank you, Kurt. Okay, let's move on to the next speaker. Is
highlighting innovations, key innovations, evidence, and their impact on patient care. So there are 4 persistent barriers to effective aerosol therapy, and they haven't changed over several decades. Those barriers include poor adherence, incorrect inhaler technique, limited visibility into medication use, and gaps in communication. Many patients miss doses either intentionally or unintentionally. In addition, aerosol therapy depends heavily on correct technique. And as we all know, improper device use can greatly reduce the amount of medication delivered to the lungs. Also, clinicians have limited insights into patients' day-to-day medication use when they use those inhalers or nebulizers at home in between clinic visits.